I must first say that election to the Israel Academy is especially meaningful to me, and I am deeply grateful. I am truly grateful. And I would like to add it's an honor that I share with my wife, Yali Lorch, in more ways than one. So Yali and I have done research on this campus of the Hebrew University for 40 years. And Yali would often say that the Hebrew University was part of her Nadunya. In fact, during the course of that research, indeed quite early on, Yali made an important finding, which I would like now to describe because I think it is both fundamental and something that I can put in language that I hope will be easily understandable and even interesting to the non-scientists in the audience. So it's a story that begins with DNA, which, as I'm sure most of you know, is the repository, the storehouse of all genetic information. The DNA must be replicated in order to be passed to succeeding generations, and that is the basis of heredity. DNA plays a second role, which is the one that is the basis of what I will tell you now, and that is in the expression of the genetic information. So gene expression is responsible for the form and the function, indeed every aspect of every living thing. Now, gene expression takes place in two steps. The first, as indicated here, the information in DNA is copied into another molecule almost identical called RNA. Because it is copied directly, the process is aptly termed transcription. DNA is retained in a special compartment of every cell called the nucleus, where it is maintained, it is protected. But the RNA, carrying the genetic message, passes outside the nucleus to the rest of every living cell, where it directs the synthesis of proteins. Proteins are the vital force. Proteins are the answer to the question that was asked at the dawn of life science about 150 years ago. What is the difference between something that is alive and something which is not? A perfect example was a yeast cell, which it was known would ferment grape juice, turning sugar into alcohol, and in the process cause the grape juice to bubble and froth extensively. It was obviously alive. And so it was asked, is this a property that is unique to something that is alive, or is it a process that is due to some internal component of a living cell? And finally, after a long time, that question was answered when yeast cells were broken open, and it was shown that the contents could ferment sugar and convert it to alcohol. And then finally, over a long period of time, the molecules that reside within the yeast cell responsible for the process were identified. They proved to be proteins. Proteins which catalyze chemical reactions. Proteins which are responsible for every aspect of life chemistry. And now we come to what is really the great question in regard to gene expression. So it turns out that every living cell contains the complete genetic information for whatever organism in which it resides. Every human cell contains the complete human genome, and yet there are about 200 different types of human cells: nerve cells, muscle cells, skin cells, blood cells. If they all contain the same information, then how do they come to differ? And the answer is that only a small part of the genetic information is expressed in any particular cell. Only proteins responsible for the properties of nerve cells expressed in those cells, but not any of the other genes, any of the other proteins responsible for, as I have mentioned, skin, muscle, what have you. So the question which has occupied research for the last 70 years and will occupy research for a long time to come is how such specificity of expression of genetic information is achieved. Now we know a part of the answer, and that is what I will tell you now. And it begins with the organization of the genomic DNA in chromosomes. The composition of chromosomes is remarkably simple. They consist of about half by weight of DNA, and the other half of the mass, 4 very small proteins, the same in almost every organism. Some time ago, as you heard in the kind introduction from Professor Dudai, I proposed that the DNA and these 4 small proteins are organized in the manner shown here. The DNA double helix in gray wrapped around the set of 4 small proteins, each portrayed here in a different color, to form a particle that has come to be known as the nucleosome. The DNA, the DNA passes from one nucleosome to the next along the entire length of the genome in an extended linear array of such particles. Now, you may be aware that the DNA of the human genome, for example, is about 100 centimeters in length, and it must reside in a nucleus of every cell that is only about 10 micrometers in diameter. The DNA then is contracted in length by about 10,000-fold. It was long thought, in fact, if you look in the textbooks or you ask anyone today, they will tell you that the purpose of the nucleosome is to package the DNA to achieve this contraction of length. They will tell you that the nucleosome is a packaging device. But the numbers argue against that idea. So in the picture that I showed before, where the DNA is wrapped around a set of proteins, the DNA length is contracted only about 5-fold. Now, it's scarcely believable that half the mass of a chromosome, almost all of the protein of a chromosome, serves the purpose of length contraction about 0.5% of the contraction of length required, ultimately, for chromosomes in living cells. It must serve a different and perhaps more important role. That came to light in work that, as I have mentioned, was done on this campus by Yali Lorch quite some years ago, when she assembled a gene in a nucleosome and discovered it would completely prevent the process of transcription. Nucleosomes repress transcription. The nucleosome is a general gene repressor. It is responsible for the lack of activity of almost all genes in all chromosomes, which leads to what I view as the first principle of gene expression. And that is summarized here. As I have said, nucleosomes repress all gene expression. Only those genes required in a particular type of cell are relieved from repression or de-repressed, and that occurs by the selective removal of nucleosomes. The mechanism by which that selective process takes place is the topic of our research at the Hebrew University today. And again, I thank the Academy and I thank you all.